Good morning, everybody. Thank you so much, Paul, for organizing the organizing committee, but also for the sweet introduction. I still remember when we met, now quite a few years ago. I thought since I'm starting our workshop, I'm going to pose kind of some challenges in my first slide, but also kind of defined protein dynamics, right? I think there's a lot of controversy, and it's mainly because of definitions. So on the left, you kind of see an illustration of an energy landscape, right? So what we really want to understand is the full energy landscape of proteins, which defines, of course, structure and dynamics. There's no structure without dynamics. There's no dynamics without structure, because what's wiggling are actually those macromolecules. And of course, beautiful, crystallography and NMR or EM methods allow us to see high resolution structures, but they are ensembles. So the first things what I want to show here that you really have to think about ensembles, and that's where the dynamic nature comes in, that they are actually wiggling around. Second, very often on covers of journals, you see the ground state, right? But it turns out, so what I'm very interested in figuring out the higher energy states, which are still minima in the energy landscape, but also then, of course, the transition state, which defines how fast the reaction goes. And sort of visualizing that, and of course, the pathways, they are transversing over those mountains. So I wrote a little perspective, which just came out last week in Nature Methods, and just to pose the challenges here. So with that said, the second thing, since we can't travel to the French Alps, I thought I'd do a virtual time travel with all of you. Hopefully we'll soon travel in person again. So we'll go 3.5 billion years back, but also we go in the future with directed evolution to ask the question, and that's really what, why can I not forward my slides? There we go. How do proteins work, and really understanding how they work. And in order to do that, we have to go beyond these static structures. And what I'm showing you here is just different areas my lab is working on. And of course, in all of those, these static structures you see here do not define function. The reason why the definition of life and the reason why those proteins function is because they move, right? And so since I was an undergrad, my big dream had been to add the fourth dimension to structural biology at the time domain and really watch those signaling molecules or these membrane transporters or enzymes doing catalysis, doing their function. So here we see a signaling protein, right? And the key for signaling is actually that this protein hops between the active and inactive conformation already before it's getting phosphorylated. And so if it wouldn't go between those two states, it could never get turned on. We were able to look in real time at a membrane transporter. It's actually a multi-drug transporter in real time, how it's actually performing its function. This will be the key player today. It's an enzyme which hopefully operates in your cells. So for enzyme catalysis, of course, these movements are absolutely essential. It's a kinase. Here we see unpublished work. It's actually the key. Hopefully this will work in your brain right now. It's the key player for long-term memory. So if you want to, hopefully remember a few things from my talk, but this interaction in your brain is essential for memory. And here we see a protein-protein interaction is actually in your eyes, and without the dynamic nature of one protein binding to the other one would actually not be binding. And of course, we are all in the same boat. We had been critiqued for a long time that, yeah, but who cares? Protein dynamics is not important for real biology or for any treating of diseases. And so I want to give you a teaser at the end of the talk to show that actually the dynamic nature of those proteins is actually essential for rational drug design. So now, of course, the question is, how do we get to these movies, and are they just scientific? the idea really is that each single experimental method will not give you the entire picture, because each method has advantages and disadvantages. But also the experimental methods alone need to be combined with computation. So that's kind of what we do in the lab. We're choosing the methods we need for visualization at different levels of spatial and time resolved, and then combine this with computation. And one new adventure, of course, is ancestral sequence reconstruction, really asking the question, how do these sophisticated proteins evolved over time? So I want to mainly introduce NMR spectroscopy for my first system. So we want to look at how these enzymes, which catalyze the reaction by 9, 10 orders of magnitude, evolved. You all remember your enzymology 100, where you're talking about an enzyme, how fast an uncatalyzed rate is. So this enzyme catalyzes the reversible interconversion between ATP and AMP to 2 AMP molecules. This is fully reversible. It's an essential enzyme in every single cell and every single organism, because it maintains the right nucleotide concentrations. So you can, if this enzyme doesn't work, you die within a minute. This reaction takes about 7,000 years without an enzyme, and is completed within 100 milliseconds with the enzyme. So this is kind of the rate acceleration which has fascinated me for many, many years. So the theme will be ask nature how it evolved this high efficiency, and then in the second part I will show you, how we as scientists, how poorly we understand enzymes, meaning that we cannot design anything today which is even close to this amazing efficiency what nature had evolved. So, usually you define an enzyme by Kcat or Km, but coming back, steering at that and steering at the static structure will not allow you to understand this enzyme. So we have to define the entire free energy landscape. And that's where solution NMR comes in, because with solution NMR, we can look at every single step in the enzymatic cycle, rate limiting step, non-rate limiting steps, and we can do it during catalysis. So the idea is we put our enzyme in the NMR tube, and as soon as we add substrate, it runs through this enzymatic cycle, and it actually runs through the enzymatic cycle forever, because it's a fully reversible enzyme. It takes substrate to product and product to substrate. So you have days or weeks to collect all of our data, right? And so how can we now figure out all these different dynamics in an enzyme? NMR is really powerful for looking at dynamics on all time scales, from picoseconds all the way to hours. All you have to do is go down here and find your right toolbox. Of course, not only that you get the entire time range, you actually get this information at atomic resolution, because you can watch every single atom in your protein. Very important, we can measure those dynamic processes at equilibrium. So it's not like in fluorescence or absorbance, where you disturb the equilibrium and you look at the return to an equilibrium to get your kinetic rate constants. And of course, we can go into physiological conditions, meaning we are in solution, we can add anything, we can change pH, buffer, and so on. So that's why it's one of the very powerful techniques. Of course, you will see also why it's tricky, why you need other methods to combine with NMR. All right, so as a little introduction, Paul asked us to define the methods a little bit. How can we measure dynamics by NMR? It's all about relaxation. And in a way, this is true for any spectroscopic techniques, right? You go in an excited state and you ask how fast you relax away. So what we do in NMR, we excite our spins. And so we go actually, you put them in the XY plane with a 90-degree pulse. And what they do now is all these spins run around the stadium with their Lerner frequency, right? So when we excite them, not only that we put them on the starting line, but they're actually all in coherence, meaning they're starting at the same time. So now they're running around, and slowly they will actually, some will run fast and some will run slower. And it's this decay, which is our transverse relaxation time. So we can now array the time, how long we let them run, basically. And so they will actually dephase more and more. So now what's the key for dynamics? If now, on top of this normal relaxation, a atom goes from one structure to the other, meaning that they actually are in different chemical environment, meaning they are running with different speeds, right? That's our frequency. They will dephase faster. So they will actually, this guy will first run slow. Now it sees a different environment, the runner will be faster. And so what you will see for all the atoms which are dynamic, meaning interconverting between structures on the milli to microsecond time scale, you will see an extra a faster decay. And it's this faster decay which we define exchange enables us to see right away this atom is experiencing a change in conformation around its environment. But we would like to get everything, right? We would like to get the populations of the interconverting species. We would like to get the interconversion rates, and wouldn't it be great to also get information about the structure of this high energy state, which might only live there for, would be there for 1% or 2%. So these methods, by the way, are sensitive enough to pick up populations to up to 0.1%. And so all we have to do is put, while they are running, while they are relaxing 180-degree pulses in the middle of this relaxation time. And this was really a very clever idea from Art Palmer now, 20 years ago. And so what we do is when we put 180-degree pulses in the middle, you're flipping the spins. So Usain Bolt, who was winning, is now last, and the last guy will be first. So it's a refocusing of the pulses, a refocusing of the relaxation, and therefore, if you give more and more pulses, we get these now famous CPMG relaxation curves. So these curves now can be fit with this equation, and what you can see, and we can measure that on every single atom. So on every single atom, we get populations, interconversion rates, and chemical shift information of the high energy state. So we basically get everything in one experiment. This is a very, very powerful experiment on this time scale. All right, so now let's do it, right? So we did this experiment on our turning over enzyme, so it's constantly catalyzing. It's old work by a team of 5 guys in my lab over the years. And so what we found that very early on, that all these red and orange residues can be globally fit with one process, with a forward reverse rate of 1600 per second, a reverse of 40 per second. Now, this cannot be the phosphoryl transfer step. I will show you in a minute, where we just, the chemical step where we just move a phosphate by 1.5 angstroms. What we found out, that we are actually not looking at the chemical step, but the opening and closing of the enzyme. Because all these residues are in a different environment in the closed state versus the open state. And we know this is a static crystal structure of the empty enzyme. That is a crystal structure of the substrates bound. So it needs to open and close. But what was very exciting to see, that the opening rate coincides with how fast the enzyme opens up. Or the opens up coincides how fast the enzyme makes ATP. So what we learned right away, that our rate limiting step, we can start drawing our energy landscape, is actually a conformational change by about 15 angstroms, the opening of the enzyme, and that chemistry is faster. And that was contrary to the literature, which described the chemistry being rate limiting. So what I want to do is not go through that, but do the new data, but leave you with a full energy landscape, meaning that we were able, over the years, and this is published in several papers here, by using room temperature crystallography, single molecule FRET experiments with my little brother, where we can, in real time, looking at the opening, closing, single turnover burst experiments, and finally, unpublished work of quantum mechanically calculations with Marcelo Marti in Buenos Aires. I leave you with the energy landscape and the movies. So here is my rate limiting step, right? The closing, here is where chemistry happens in the closed state. And the rate limiting step is this opening of the ATP and the AMP lid. And it, of course, needs to open to release the products. For this audience, I wanted to really show one other important question. Everybody focuses in enzymology on the reaction which needs to be catalyzed, which means the phosphoryl transfer, which I show you over here, right? So what you see over here is really how you start from ADP and make ATP AMP by going to the transition state over here, right? And I think you can appreciate how little that phosphate has to move, meaning if you're really nicely organized in that active site, the work has been done. so that chemistry becomes very fast, faster than the opening. So everybody focus on that reaction, but in fact, the hardest thing is to suppress the unwanted reaction. And think about what the unwanted favorable reaction is. It's actually hydrolysis. We are in 55 molar water. And this is much harder, right? And if this enzyme would have any hydrolysis, you would be dead in a minute, because if your ATP concentration drops by 1 millimolar or 2 millimolar in your cell, you cannot survive. So any hydrolysis where you just get rid of, you hydrolyze your ATP would be detrimental. So the enzyme had to work really hard to suppress that. And so I naively thought it's going to strip out all the waters from the active site, but it's not. I show you in long and decimylation here, and all these balls are water molecules, and the structured water molecules are shown as H2O here, which we see in the crystal structure. And what you see is it's full of waters, right? But the waters don't escape the cavity, number one. And number two, they are held far enough in place that they can never do nucleophilic attack. So these closest water molecules, or really, we see them in every crystal structure, in an end-decimylation cannot move close enough to the phosphates. Okay, so now that you have kind of an idea how this enzyme works, right, with these different microscopic steps, we can ask the question, how did it evolve? And if you ask a question of evolution, you have to ask, what is the evolutionary driver? Okay, first of all, evolution only works on organismal fitness, or not on a single enzyme, but it's going to be apparent why this works on a single enzyme, because this is, first of all, essential, and second, the speed of this enzyme is actually essential for how fast the cells grow. So I was thinking about what is a very strong evolutionary driver over 3.5 billion years. And I came up with temperature, because of course, the key for this wiggling, and therefore the key for any biological function is KT, is temperature, right? The energy comes actually from external temperature. Now, why is it such a great evolutionary driver? You see the dilemma. When nature arose, when basically life arose about 3.5 billion years ago, the Earth was a volcano. So we had plenty of energy, right? So tons of energy for these chemical reactions. But of course, over the years, the Earth cooled down a lot. So in other words, our enzymes today need to be as efficient with much less energy as the old enzymes. So they actually have to become more efficient, right? And that's a very big temperature gradient. So while we were doing these reactions in the lab, we had a conference close to Yellowstone. I visited Yellowstone, and I saw these hot vents. And it turns out this evolution actually happens still today. So this temperature gradient, from volcano in the middle all the way to the outside where it's cold, you see in these hot vents. And so the color coding is actually the different microorganisms which have different temperature optima. So I thought it was kind of a cool analogy. If you haven't been to Yellowstone, go there. It's really, really fantastic. All right, so we have a question. We need the theory. Okay, the theories we have in theoretical ancestral protein. Here's my activity temperature profile. Everybody has seen that. And so the theory is it's very little active at low temperatures because it was hot back then. And then, of course, normally goes up, as we all know, with temperature. And at some point, the activity goes down purely because the protein unfolds. Now, what happens when we go from hot to cold? The evolutionary pressure is I need to get active, more active at lower temperatures if I want to survive as an organism. So you will go up that's a theory that you increase the activity at lower temperatures. But look what happens if you go the opposite. If the temperature goes from cold to hot, it actually happened in evolution. About 1 billion years ago, Earth froze over. It's called the snowball effect. And we will look at that, what happens in evolution there. What's the evolutionary driver? Is it activity? It's not. It's already active enough at lower temperatures. So it's just going to get more active at high temperatures. But what is the evolutionary driver now? It's stability, right? Because you do not want to unfold. So what you see with a very simple thought experiment of going from hot to cold or cold to hot as evolutionary drivers, you have two physical parameters to dial on your protein, activity in one hand, stability on the other. All right, now we have a theory. now we actually got to do it. How do we actually ask the question how these enzymes looked like all the way back when life arose on Earth? And that it only has become possible because of the genomics, that we have so many modern sequences now, right? Which we can look at and then calculate first a phylogenetic tree, which I'm sure everybody has done, right? So we've started a phylogenetic tree, but second, we are using those sequences together with an amino acid substitution matrix, basically how fast amino acids are mutating, physically probabilities of sequences on these nodes. And again, it's a probability calculation, right? So each amino acids will get just a probability what it was back then. Then finally, we order the genes and make those proteins. Of course, most importantly, you have to hire 2 brilliant Homo sapiens to execute that research. And these are my superheroes, V and Chris in the lab. So the first thing I'm showing you is just stability, melting temperatures. The temperature number, the melting temperature is in numbers and color coded here, meaning that the old ancestor is indeed very thermostable. And if you follow the melting temperature as a function of time, what you see, it follows really nicely the temperature trend of the sea temperature on Earth. And you see about 1 billion years ago, that ancestral protein was the least thermostable one, and that was exactly when the Earth froze over at this ice time. Okay, but temperature, thermostability is easy to do. What happens with activity, right? Remember, we have to make our enzymes faster as we cool the Earth down, and this indeed happened. Here's my old ancestor activity temperature, and now I show you a younger ancestor tree. Indeed, it became more active at low temperatures. So yes, our theory actually is correct, right? And they had to become more active. Now I show you all the ancestor proteins. Of course, that's a lot of work for me. In the more familiar Eyring plot, where we plot the ln of the rate constant versus 1/t. So in other words, the low temperature is over here. And what you see is that the ancestral proteins are really little active at low temperatures, and they become indeed more active to the modern one. But they become more active because we have a strong curved Eyring plot. So now, if you remember your physical chemistry, those plots should be linear, right? Because the slope should be the enthalpy of activation and the intercept should be the entropy of activation. So why are they curved? That is actually violating theory. So I thought I have the answer because I remember my enzymology class I took. And of course, when can you get a curved Eyring plot? It's when you have a change in the rate limiting step. We know for the modern ones, the opening rate is rate limiting. If the opening rate has this temperature dependence, but the phosphotransfer step has a different temperature dependence, what you get is a crossover that at low temperatures, a different step, meaning the phosphotransfer, can become rate limiting. So I thought, that's what's going to happen. Except my postdocs convinced me otherwise. At all temperatures measured by these NMR relaxation experiments, the rate limiting step, that's really low temperature, is still the opening. And second, you can do a single turnover enzymology experiment where we ask the question, how fast do we make ATP? And we see on all temperatures a burst. The burst is basically the phosphotransfer, which is fast. It's too fast to be measured. It's in the dead time of the experiment. We turn over as much ATP as we have enzyme. And then the linear phase is actually the slower rate limiting step. All right, so we were stuck. It's like, what are we going to do? But I remembered my PChem. I had a really good professor when I was in, a sophomore. And there is this thing which is called the heat capacity. Of course, in this audience, you will be hopefully familiar with heat capacity, right? The heat capacity of proteins. It's a reason why an egg boils, right? Because the denatured state and the folded state of a protein actually has a different heat capacity. And it's a negative heat capacity. So your breakfast egg boils because the heat capacity is smaller for the denatured state. We are assuming, so this has been very much applied for protein folding, but not for enzyme catalysis. what we always assume that the heat capacity of the transition state is the same as a ground state. But since my transition state for this opening is actually kind of a protein folding thing, right? It's going from a closed to an open and so many atoms are involved. I mean, we should not assume that the transition state heat capacity is the same as a ground state. So if we actually say that the heat capacity is different, you just expand the iron equation with a delta Cp, and that gives you a curved iron plot. So we can explain all of our data by a difference in heat capacity, where the heat capacity of the transition state is smaller than the ground state. And so these curved iron plots become straight in evolution, and therefore the enzyme becomes faster by a change of the heat capacity of the difference between ground state and transition state. I will come back to that because that's a new theory. Before we go there, let's go now cold. This was hot to cold. By the way, I wanted to emphasize that I show on the structures in black dots all the residues which change in amino acid from ancestor to ancestor, that you can see how many physically changes of amino acids we get along the evolution. Now, this is that cold guy, remember? Now, this creature was heat-loving and went to these hot vents and became a modern thermophile. So now we go from cold to hot, and I told you the theory is that our evolutionary driver is stability, and indeed, this enzyme became more thermostable. But what happened to activity? Remember, it's already very active at low temperatures. There is no change in activity. It stays very active at low temperatures, right? So the only thing which happens, it changes is stability, and we come back to that. It's not going to get curved again to become little active at low temperatures. And just think about for a second why, while I'm giving you one more data set. Since we came up with this really weird thing of biophysics, heat capacity of the transition state as a reason why these evolutionary things are so different, I thought we need a strong experimental control of that theory. And we went back to our phylogenetic tree. And there are two thermophilic enzymes which actually never went to a cold intermediate. So these were microorganisms which stayed in the hot vents the entire time and never experienced this cooling down on the earth. So I'm asking the question, how should those enzymes look like? Did they ever have an evolutionary drive with respect to temperature? They didn't. So in fact, they should just look identical to these old ancestors, right? Because nothing had changed over 3.5 billion years. However, half of the protein has changed an amino acid sequence. However, all the physical parameters, stability and activity, temperature profiles have not changed at all. And why? They never had the pressure to become more active at low temperatures. So what is going on here? This is just evolutionary drift. These are amino acid changes which do not affect at all the function of this protein. It's just neutral drift. Now, the other thing, what we can do with this ancestral sequence reconstruction, actually answer the question why modern creatures are different. So you see these two thermophiles, and people have thought forever why they are different. One is very little active at low temperatures, the other one is highly active. Now you see why. It's the evolutionary history. So if we want to understand function of modern proteins, we have to understand their evolutionary history. It's still in there today. The reason why this guy is active at high temperature is because it had this evolutionary pressure during evolution to be active at high temperatures. All right, so which leads me now to the general take-home messages or things we learned about principles about evolution. The principles about evolution, I call it here active pressure versus passive drift. Okay, active pressure, remember, was the enzyme has to become more active at low temperatures, and that's fast. If it doesn't get more active, this creature will die. But it did not lose its thermostability. That's weird, right? Because you learned that if you make faster enzymes, you can only do that by making it less stable. So first of all, that theory is wrong. So this is stability and activity trade-off. Second, I call that a super enzyme. because it's super active and it's super thermostable. Why is it? It's because there is no disadvantage to be thermostable. So there is no active pressure. I have to become less active. But where is this paradigm, which you all learned about the flexibility stability trade-off coming from? It's coming from looking at modern enzymes. So let's follow our evolutionary time travel. Over time, indeed, adenylate kinase is losing its thermostability, but it's slow. And it's passive drift. It's slow because it's passive, because there's no disadvantage. But there's also no advantage to be thermostable. So meaning there's no purification. So by chance, it's going to lose it. And that's where it comes. If you look at the modern mesophile and the modern thermophile, that trend is usually there, but it's only a result of passive drift. Since I made you a super enzyme, I want to make you a super organism. Why super organism? This creature, the purple one, was really puzzling to the microbiologists because it survives a very wide temperature range. We know why it's surviving a high temperature range because of thermostability. But most thermophilic organisms cannot survive low temperatures. And remember, I told you, this is a creature where the activity of this enzyme is still high at low temperatures because it hadn't lost it yet. So I made a very bold prediction. We put in the last sentence of the paper, and the editor wanted us to take it out. I made a very bold prediction that it's the activity of the little enzyme of adenylate kinase, the speed of that, which allows the full organism to survive. Luckily, my grad student, Chris Wilson, bailed me out with new experiments, which are not published, to prove that, that the enzymatic speed of this enzyme controls the organismal fitness. So the theory was here, I showed you, right? That if the enzyme, like this ancestral protein or this really slow modern protein, if it's below a speed limit, right over here, the cells will not survive, right? And I'm German, I love to speed, so it's a minimum speed limit, not a maximum speed limit. Anything above, it will be fine. And so what Chris did, he knocked out the genomic one and put in those slow enzymes or the fast enzyme in E. coli and measured the growth rate. And in exactly what we predicted, below the speed limit, the enzymes which are below this magic speed limit of 60 per second, the cells barely grow. Once they hit the speed limit, and remember, they're still much slower than the E. coli enzyme, they grow as fast. So it's a minimal speed limit. Now, of course, now having this direct link of the speed of an enzyme to organismal fitness, we can now do directed evolution. And so what my guys here, this is the new team, built was a whole bit turbidostat, where we can do directed evolution in real time at these low temperatures. And indeed, it works like charm, right? So the evolved ancestor we to put in a library with many variants, and the evolved enzymes have a faster doubling time, right? The organism grows faster and they have a faster KCAT, and we're in the process of analyzing all these data. So that's our new team in the lab. So we can indeed use organismal fitness of the speed of this enzyme to do directed evolution. Okay, so this was kind of going around the evolution. The second big question, which had bothered me for a long time, is looking at the transition state. And of course, we cannot visualize the transition state directly with conventional structural biology, because the probability to be up there is maybe 10 to the power minus 9. So whenever you read papers that by EM we can look at a transition state, I hope your cross is very much off as a red cross. So how can we view the summit, right, if we can't view it?
Make sure that she's got a little bit
so
of gap, and she's spreading
that's what we want to do. We want to look how the protein goes over the transition state. So I can watch my little daughter racing at the World Cup by just turning on the TV, but how do we do it microscopically, right? So microscopically, of course, this is the audience to talk to. We do MD simulations. That's the amazing thing with computation. We can watch it, right? But the issue is we did the target MD, we had to do targeted because it's too slow for even brute force on Anton. We don't know along this pathway where the transition state is. Okay, so that's the question we want to do, right? See, along this, I mean, first of all, is this pathway realistic, and where along is actually the height of the energy? So I came up with an experimental indirect way to visualize the transition state. And here's the idea. What we can measure are rate constants, right? So if we can measure rate constants, and we can measure how fast this enzyme goes over, right? So there's an indirect way of this rate constant directly related to the transition state. Now, I came up with the idea to measure the pressure dependence of this opening, okay? Why pressure? Because it gives us information about the difference in partial mole volume from the ground state to the transition state, okay? Now, we needed to do a second trick. We had to compare two homologous enzymes, and I'll show you in a second why. So now, don't time travel with me, but dive down 10 kilometers into the ocean, find a creature, this mesophilic profundum, which lives in the bottom of the ocean under high pressure. So we're going to look at adenylate kinase from this creature and from E. coli. So the first thing what Jordan did, we measured the activity as a function of pressure. So we built a little device where we can turn by hand the pressure on our little spectrophotometer and measure the activity by an enzymatic assay, because remember the rate limiting step is this opening, so we can just measure how fast we make ATP. The mesophile couldn't care less about different pressures. So these are the pressures we use, right? These are the raw data, no pressure dependence on this rate constant. In contrast, the piezophile starts really, really slow at low pressures and becomes much, much faster at high pressures. So that was a good day, right? We have a strong pressure dependence for the piezophile, the pressure enzyme. Then we did, at atomic resolution, the dynamics of these two enzymes at different pressures. So we need a pressure tube in our NMR tube, which was actually developed by Arnold Patterson. They are now commercially available. You put it in, you turn on the pressure, and you see the data look absolutely gorgeous. Jordan, John, and René did these experiments. Now we cannot measure dynamics, right? We have now 3 dimensions. These are our normal relaxation profiles as a function of pressure. And you can globally fit all these data, and what comes out is actually the rate constant at low bar and high bar. And then, importantly, we get the partial mole volume difference between the ground state, which is a closed state, and a transition state, okay? So what you see here is that indeed the piezophile has a large partial mole volume difference. And what does it mean if a reaction gets faster with pressure? If it gets faster with pressure, it actually means that your transition state is smaller in partial mole volume than the ground state, because you're making the reaction faster, right, going to the transition state. And so this difference is 35 mils per mole versus the E. coli is basically no difference in partial mole volume. Now, you said I wanted to give you high resolution structural information about the transition state, but I just give you the volume, which is just a blob. We have to ask 2 more questions. One question is, what defines the volume of a protein? It's actually not, you can barely squeeze a protein, but what you can squeeze much more is water molecules around charges. So one residue is becoming charged gives you about 10 mils per mole in partial mole volume. So when we saw these data, we predicted that the piezophile will solvate 3 to 4 charges in the transition state relative to the ground state, whereas the mesophile will not. And that's where it's kind of the idea. We're doing a comparison. We're going back to our molecular dynamics simulations and ask the question, where along the pathway do we see solvation happening on charged residues, right? And it should be on the right side for the piezophile and should not be for the mesophile. That's why we needed the 2 homologous proteins. First, I show you the MD simulation here, and you see the water molecule zipping in. That's the other great thing about MD simulation. You can actually see the water molecules, and you indeed see how the solvation happens in real time. I'm freezing those frames right where we think the transition state ensemble is. This is the ground state. right? No water molecules in there, right? In this interface. And now very early on, just when the two lids are starting to open, in the piezophile, we get this solvation. And in the mesophile, in the E. coli, we don't. And why? Because we have a strong salt bridge between the aspartate and the lysine over here, which we don't have over here because we are swapping two residues. So this lysine becomes a glutamate, okay? And therefore, we cannot make that salt bridge. Now you see where I'm going, right? We had experiments, now we have computation. The computation makes a very strong prediction. If this is a transition state, we can actually experimentally test it, right? How would you test it? The prediction is, if this is a transition state and we put those two residues into the piezophile, it should become pressure dependent. So that's how we do it between experiments, right? Computation allows you to make very much strong predictions. So what Jordan did, he put those two residues into piezophilic background. Of course, the piezophile has 50 different residues, so you're only swapping two. And indeed, what he found that this double mutant has 30% of the pressure dependence. That's not 100%. So what happened? We missed one other player. So when you turn around that interface, on the backside, there was one other residue which we seem to be crucial for this transition state opening for solvation. And it's this glutamate, which doesn't allow the opening because of a strong bridge to this over here to the backbone and to this lysine. This becomes a valine. So now we put the valine as a third residue into the mesophile and get the full pressure dependence of how we see it in the mesophile. So remember, just pause for a second. You take one whole protein and just put 3 residues, which were predicted by computation, into this mesophile, and we recover the full pressure dependence. So we were able to bracket, and again, a transition state ensemble, it's not a single state. We were able to bracket roughly where the transition state is, and it's really very early on, much earlier than what computational papers predicted based on pure computation. As soon as it starts to open, that's the transition state ensemble for this enzyme. Okay. This is a natural evolved enzyme. Can we take those lessons for design to improve the enzyme design? This was really beautiful work as a collaboration with Don Hilbert and his grad student Adrian. So we basically want to ask the question: why are the designer enzymes so lousy and become so much better with directed evolution? And some of you might be familiar with that story. It's the catalyst which catalysis reaction, a breakage of this ring going through this transition state, and this transition state can be very much simulated with a transition state analog. The designer enzyme is really lousy. It's only 3 per second. It was designed by Steve Mayo, and then Don Hilbert in this paper 2013 evolved it and became much faster. And when they solved the crystal structures, they basically looked the same. So the crystal structures were really misleading. But there was one thing they saw. This is always the evolved one, the 317, right? Here's my transition state analog. And of course, what was designed, the catalytic base and acid to do those base acid catalysis. And this glutamine, this lysine did not interact well with the transition state, but the evolved one put a glutamine in. So of course the idea is, oh, all we have to do is put this glutamine in, and that's what we actually did as a first experiment, because that should work perfectly. Put the glutamine in the designer one, it looks perfect based on the static crystal structure. This should just be as active as the evolved one, which by the way, put in 17 residues. However, it's basically not faster than the old one, right? So what's going on? It's very misleading. It should be as active as the evolved one, it's not. So we turn to NMR, must be the dynamics. And so we found that something weird. When we took NMR spectra at higher temperatures and in higher pHs, we actually saw 2 peaks per residue. And what we later on, basically showed that it's an active and inactive state. So there are 2 states which are both folded, which interconvert slow on the NMR timescale. And that's the reason why the evolved one also loses activity very early on, because we're increasing this inactive population. Now, looking at the Zymer enzyme, it turns out that this inactive state is there to a pretty high percentage to start with, okay? We could measure in real time the interconversion rate. So you see the inactive gets less and less populated by a jump experiment. And so the interconversion time is actually in hours, okay? Or like in 30 minutes. So the first thing, what we envisioned is that we have an inactive and active conformation, which actually now solving the crystal structures without the transition state analog bound, we could directly detect in the crystal. So we have an inactive state and an active state, and the inactive state in blue actually does a steric clash with the transition state. So in other words, you cannot even bind the substrate. And the first advantage with directed evolution that we are kind of losing more and more this inactive state in the evolved one, okay? So that's the first one. Now, I told you that in the fully evolved one, the inactive state is still present, but it's very low percentage at normal temperatures. So we wanted to capture this inactive state for the evolved one. And also in these crystals, the inactive state was kind of localized in the active side. So what Ricardo designed was a new device where we can do crystallography at really high temperatures, at 70 degrees. And at 70 degrees, we could directly detect for the evolved one the inactive and active conformation. And it's a massive conformational change. Several backbones have really flipped fully, right? And so I just wanted to put this in because it's kind of a cool technology now to go to really high temperature. In these crystals, we were able to actually allow this flip to happen. Now the dilemma is that this shift in population only gives a 3-fold increase in overall activity, okay? So where is the other 200-fold coming from? So this is our conformational selection, right? Active, inactive gives us a 3-fold difference. The rest, the reason comes actually from the chemical step. And how can we test the chemical step? Remember that transition state theory tells us that an enzyme has a higher affinity to the transition state relative to the ground state. So if we can just measure the affinity of these different enzymes to the transition state, that should basically detect our rate acceleration, right? And indeed, that's what we see. There's a perfect correlation between the increase in affinity to the transition state as we evolve to the rate acceleration, which means that this transition state is a perfect mimic. Now we can look at the crystal structure of this transition state. Remember, I did this early on, but we looked at static structures. What I'm showing you now is a really cool method, which was published in 2012, and I'm sure Jamie Fraser will talk more about, is ensemble refinement. So you're ensemble refining your crystallographic data together with an MD force field. And now you see what happens. Remember, this was the one which we thought should be perfectly as fast, but it's not. And the reason is because we have lots of wiggling around. So the catalytically active form is in there, but it's a low probability. So in other words, what directed evolution does to find the really fast one, it minimizes unproductive fluctuations. And it minimizes unproductive fluctuations so that you're now increasing the rate of the real reaction, right? So you see how this glutamine and this aspartate becomes very well defined, and it's all over the place here. So the last thing what Ricardo did, and that was really clever, he looked at this and says, we don't need those 17 mutations. I think this methionine is a bad player, because it gets in the way of this favorable interaction of this methionine to the tryptophan. It said, I predict, based on these data, I put, instead of the methionine, the cysteine in, and it will become active. And boom, the major activity we get just with this single mutation, okay? So pure on these data, right, on this ensemble refinement, we could design basically which residue was a key player, and the other ones play a very, very small role, right? Which is good news for enzyme design. So, you know, it is kind of defined in one shell, kind of where we can make substitutions to make it very fast. So, I should stop there, right? Time-wise? Yes.
Time-wise, to leave time for questions.
Yes.
Maybe you have the last message.
Yeah. So exactly. So maybe just 1 minute for the teaser, how we can use that information for rational drug design. You all know that protein kinases are essential. All I want to leave you at that the key for that Gleevec, and these are just the 2 papers, I didn't want to talk about it anyway. The key why this drug is so selective and only really binds very well to Abl kinase with nanomolar affinity and a 3,000-fold less affinity to homologous proteins is pure protein dynamics in the drug-bound state over here. It has nothing to do with binding. It has nothing to do with conformational selection. It's really what I show you as a wiggling. It's really dynamics in the drug-bound state, which we call induced fit step. And I'll leave you with why allostery is important. This is the other 2 papers you maybe want to read. The idea is not to do active site binders, but take advantage of this dynamics, the allosteric network in a protein here, our kinase, where we have an allosteric binder over here, and where we can take advantage of this whole allosteric network and design binders which bind to this allosteric site. In this case, we used monobodies, and just to show that those monobodies which bind over here, in activating monobody and an inhibiting monobody, can either activate a protein or inhibit a protein. So finding binders to these allosteric sites, we can dial in at will activation or inhibition by either small molecules, or in this case, monobodies, which are 100% selective. And again, taking advantage of the allostery to make drugs. So just to leave you with that, right? Not rigid body docking, but rather taking advantage of protein dynamics in the apoprotein in the drug-bound state to make very selective drugs. That's sort of the outside, and that's what we do with Relay, right? So I hope I've shown you that nature really evolved the free energy landscapes, and all we have to do is to exploit those natural dynamics for allosteric or orthosteric sites, right? This is the important part. This is the basic research. This over there is now going on at Relay Therapeutics and MoMA Therapeutics, but it's all based on basic research and academics. The most important slide here is my team, amazing group of people, which really produced all the data here, and I'm so proud of them, and I just have so much fun working with them. Thank you.